Good afternoon. It's my great pleasure to introduce today's speaker, Professor Volker HAUCKE. So he is a full professor of the molecular pharmacology at Freie University of Berlin and director of the Leibniz-Institute of the Molecular and Pharmacology at Berlin. So Professor Volker HAUCKE received a degree in the biochemistry at the Freie University of Berlin, and then he moved to Switzerland, right? And he received a PhD in biochemistry in University of Basel in 1997. Then he moved to Yale. Then he worked with Pietro di Camilli. He's one of the very famous synaptic vesicle exocytosis and endocytosis. And then he worked there for 3 years, and then he moved back to Germany. He became an independent research group leader at the University of Göttingen. And 2003, he became a chair of the Department of Membrane Biochemistry at the Freie University of Berlin. And 2012, he became a director of the current institute. And then he did active work. So he did a lot of work about the membrane and synaptic vesicle and vesicle exocytosis and endocytosis. And then he published also numerous papers in high-profile journal. Like every year, he published one paper in Nature or Science or Cell. I would like to know the secret how he can publish the high-profile journal in every year. Anyway, so because of his outstanding work, he received many prizes. In 2016, he received the Reinhard Kosselhock Award of the Deutschland I call the DFG award. Okay, I'm sorry, I cannot pronounce the German very well. And then he received another important award. This is quite an important award, the Avante Award in the Lipid of the American Society of the Biochemistry and Molecular Biology. Actually, this is a pretty prestigious award for the membrane science. And then also he received the Friedberg Award for research in physiology and pharmacology in 2020. And then he was elected, I would say, several membership. So he was elected member of the Academy of Europe, and also he was elected of the German National Academy of Science. And then in this year, so he is the president of the German Society of Biochemistry and Molecular Biology. He has done a lot of work, but today, so he will present about the repeat control of the membrane dynamics in health and disease. Without further ado, please join in me in welcoming the Professor Volker HAUCKE.
Thank you very much, Hyo-kyun, for the most kind introduction. And thank you all for being with us this afternoon. It's a great pleasure to be in Hong Kong. And actually, it's my first time in Hong Kong and to present some of the work that we've been recently doing to this audience. And what I would like to do this afternoon is to give you an overview of two recent stories, more or less, that have to do with lipids. And I suspect that many of you came because you're so kind, but you may not actually be too interested in lipids. So I hope that I have a chance to change this view this afternoon, at least a little bit, by getting you excited about the roles of lipids in membrane dynamics. And as you will hopefully see, that role pertains to many physiological functions, ranging from metabolic disease to muscle disease, to basic scientific understanding of how cells and tissue responds to nutrients, to eventually brain function. So let me start out by introducing you to the roles of membranes and membrane dynamics in nutrition and metabolism. Metabolism, of course, is something that you all care about. Even if you're not actively researching metabolism, you're experiencing that every day, and I can admit that I've been so well fed in this place. We've had wonderful meals thanks to our wonderful host, and it's been a great pleasure to be here. Now, what all that wonderful food does is essentially it's making us happy, but it's also causing problems, because being overfed is one of the most prominent problems of our modern societies, with effects that make you prone to suffer from diabetes, hypertension, atherosclerosis, and other types of cardiovascular disease that are related to changes in metabolism that are induced by being overfed. So understanding how cells and tissues, and in fact our entire body, responds to nutrients and nutrient supply is pivotal to understanding these diseases. If you followed the field of cancer, you might have actually seen that drugs that interfere with growth-promoting pathways very much depend in their effect in human patients on the diet that these patients get. So it's really worth dealing with this. Now, metabolism and metabolic signaling also, and by and large, is affected and impacts on aging. And that is evidenced by the fact that many of the diseases that you've just seen, in fact, are related often to occurrence in late stages of our life. Many of these cardiovascular diseases, hypertension, age-related diabetic forms are related to aging, and in fact typically impinge on regulatory pathways that control metabolism at the level of changes between catabolic processes such as autophagy and the biogenesis of lysosomes, which I will tell you more about, versus growth-promoting signaling pathways. And these, in most of our cells and tissues, are regulated and controlled via nutrients through a complex that is called the mTOR complex, that I will tell you about more as we progress through this talk. So just keep in mind for now that these dietary restrictions and interventions, in fact, at the end of the day, typically impinge on catabolic processes and the regulation of those processes by nutrient signaling complexes, including, but not only, involving mTOR. So the question that I would like to get you excited about now is, in fact, this question. How does a cell know about whether you ate a beautiful lunch or dinner, or doesn't know, and how does it react? In fact, if you open a textbook of biochemistry, you will find all sorts of changes that may occur if you interrogate metabolism, but it typically doesn't answer the question of where this happens, because in the individual cells of our body, there is many organelles that in fact are responsive to metabolic cues, and there's a lot of things that need to be coordinately changed rather than stand-alone responses of a particular pathway. So we're very interested in understanding how organelles really respond and coordinate to metabolic cues. Let me list you some of those, and I'm sure you are aware of most of this. When you don't eat, if you're not in Hong Kong and you're being hosted by Professor Park, then in fact, you're driving lipolysis, so all your fat tissues get burned, and that you might witness as a good thing. Autophagy gets induced if you don't eat. Lysosome biogenesis gets induced. Lysosomes, just to recap, are the end point of the autophagy pathway, because the autophagosomes eventually fuse with the degradative lysosome to resupply the cell with nutrients that are liberated through the activity of acid hydrolases in the lumen of the lysosome. Mitochondria undergo fusion and in fact get prominent roles in lipolysis, because they carry out the beta oxidation that is eating up the fatty acids that are liberated through lipolysis and probably supplied by autophagy, while protein synthesis and any anabolic processes, including cell growth and division, are downregulated. So a couple of years back, we've started to embark on understanding how these various responses that you can see only some glimpses of are really coordinated on a cell-wide scale across different organelles. And since I don't want to dive deep into published stories, let me just tell you that one element of what we found was that when cells undergo amino acid starvation, it is not only that you start to induce autophagy, but in fact organelles that were perhaps not on your personal or my personal roadmap of metabolically sensitive organelles, the endoplasmic reticulum undergoes a drastic change in shape. The endoplasmic reticulum, as you may recall, seals the nuclear envelope and is comprised of ER sheets that are covered with ribosomes that make all the secretory proteins that David Banfield and colleagues study. It is also comprising smooth ER tubules that are the sites of calcium storage and signaling and of lipid biosynthesis. And these are continuous organelles that are linked throughout the entire cell. Now, when cells undergo starvation, as you can see here, the ER labeled by Sec61 in live super-resolution microscopy. When cells undergo amino acid starvation, this peripheral tubular ER network gets converted into sheets. Now, the purpose of that change, no, it doesn't change anymore, okay. The purpose of that change and the mechanism of action, and I don't have time to tell you the data that underlie this, are in fact effecting at the end of the day a fusion of mitochondria into larger organelles that are responsive to receiving fatty acids that are derived from the lipolysis of lipid droplets that get carried out in beta oxidation through an endothelin signaling pathway in which a particular lipid, PI3P, gets hydrolyzed from endosomes, and this endosome that normally is linked to the tubular ER in the periphery lets go of those ER tubules. The tubules snap back, and the tubular ER in the periphery of the cell is converted into sheets. Now, sheet ER, in difference to tubular ER, is unable to promote mitochondrial fission. So a response that is initiated at an endosome, an organelle that receives signals from things like insulin receptor, is transmitted via a change at the level of the morphology of the endoplasmic reticulum to a response at mitochondria. So this is just to illustrate to you how dynamic cell changes occur in response to different supply of various kinds of nutrients, just as the sort of starter menu. What I would like to tell you now about is a very recent study in which we've actually asked a question that cell biologists and biochemists have been interested in for a long time, that in fact have been noted for a long time, and that is the question how lipid signals really now control the transition between the growth-promoting fed state and the hungry state at the level of an organelle that is called the lysosome. Let me explain. So a little bit about the lysosome. You've all heard about lysosomes from basic courses in biology and cell biology. The lysosome is an organelle that is involved in several different things. You will probably all remember that the lysosome is an acidic organelle that in its lumen carries acid hydrolases that execute cellular clearance of material delivered to them, either through the digestion of internal proteins through autophagy or extracellular material that is derived from nutrients. A great example of that is cholesterol that is, in fact, endocytosed and eventually delivered to the lysosome from where it is reshuffled to the endoplasmic reticulum. Now, the lysosome, as it has become clear in recent years, is also the major control site for cellular metabolism. And that is because the lysosome is the site of action, or so most people believe, of the mTORC signaling complex. This is a complex discovered originally in yeast, but present in all of your cells and tissues. That is one of the major nutrient sensing platforms that integrates input from growth factors, amino acids, and glucose for the cell to decide whether it's in a growth-promoting, divisionally active, or in a catabolic state by, in fact, integrating these signals and directing them to the lysosome to change the fate of action. Now, lysosomes and the importance of lysosomes is also witnessed by the fact that many human diseases, and I'm only listing very few here. In fact, there is a long list of lysosomal storage disorders in which individual enzymes of the lysosome or enzymes that indirectly affect lysosomal catabolism are defective, and you can see the effect essentially every organ of your body, most notably the central nervous system. So this is just to convince you that the lysosome perhaps is an organelle worth thinking about if you're concerned with questions of biology of metabolism. So in essence, the question that Michael Ebner, a postdoc in the lab, had been asking when he came to the lab 5 years ago was essentially to understand how the lysosome can do two things that are apparently contradictory to one another. On the one hand, the lysosome is known as a catabolic organelle that degrades macromolecules because in the acidic lumen of that lysosome, it degrades material that goes in, lipids, proteins, carbohydrates. For that, it needs to be acidic and nothing else. On the other hand, it has been now shown that the lysosome, through work from David Sabatini and Mike Hall and others, that the lysosome in an anabolic growth promoting state is the site of integration of these various factors such as insulin and growth factors and amino acids to direct anabolic signaling, which does the exact opposite of catabolism because mTORC is repressing the catabolic activity, it's repressing autophagy, while at the same time promoting cell growth and secretion. So how can the same organelle, on the one hand, promote growth and repress catabolism, while at the other hand, promoting catabolism? That seems like a no-go. Is there different kinds of lysosomes? Is there something going on with lysosomes? That was the question. So in principle, you could come up with two different scenarios, right? So what happens to a cell and to the lysosome when you don't eat for the rest of this week? So in principle, you could come up with two different scenarios. Number one is, well, I don't eat, I make new lysosomes that are really catabolically active. Those are the guys that now in starvation give you all the goodies that you eventually need to promote degradation of macromolecules and to refeed and keep your cells alive. Or on the other hand, you take the lysosomes that you have and you do something to them miraculously that will stop the growth promoting activity of that organelle and convert the existing lysosome into a catabolic organelle. So we asked that question, and in fact, Michael conducted a series of relatively simple experiments. You take cells of your choice, and these are, in this case, standard cell lines, osteosarcoma cells. You withdraw amino acids and growth factors, and you ask what happens to the lysosome. And some of this is actually an old hat. If you followed the field, you will recognize that some of this has been known. So in a starved cell, lysosomes, labeled here by LAMP1, become perinuclearly clustered near the cell nucleus. They also become more acidic. I don't show you the data. And they ramp up the catabolic activity. This is an active probe that reads out how active the proteases inside the lysosome are, and they're much more active in the starved lysosome than in the fed lysosome, okay? Now, if you then stop protein synthesis through cyclohexamide, so you're blocking the de novo pathway, that was the one option on the left, right? And ask, does that still happen? The answer is yes, it doesn't care. I don't need to make any protein to ramp up the catabolic activity of the starved lysosome. Now, if you don't believe this, we didn't believe it, you can actually go to proteomics. You can take isolated lysosomes and do quantitative proteomics of the lysosomes from these cells. Now, what you see plotted here are all of the lysosomal cathepsin-like proteases, of which there are many, about a dozen. And this is the log difference between the starved and the fed, and you can see it's a line in the middle. There is not a single change in the lysosomal proteome at all. So the organelle is exactly the same. However, it does become proteolytically very active. So what happens to protein secretion? I said when a cell grows and signals mTOR is active, it secretes proteins. So we used a simple established readout, a vesicular stomatitis virus G protein, which you can accumulate at the level of the Golgi complex and then chase it out through a temperature shift from the ER-Golgi system to the plasma membrane. And if you do that, then it will eventually, these are fairly flat cells, it will land at the plasma membrane and in these vesicles that are about to reach the plasma membrane. There's still some in the Golgi in this case. If you do the same experiment in a starved cell, you find, in fact, that the VSVG is retained at the level of the Golgi. So protein secretion seems to be stopping at the level of the Golgi, while lysosomes become catabolically more active. So what is the reason? Is there perhaps a connection between these? Now you got the Golgi and secretion, and you got a change at the level of the lysosome. Is there anybody who is the mastermind that controls this? And here is where I want to get you excited about lipids. Phosphoinositides or or membrane lipids that occur in all of your cells. They're a minor lipid, and they're in fact quite boring when it comes to their basic structure, which is a phosphoglycerol backbone with 2 acyl chains, typically 1 unsaturated, and an alcoholic head group that is inositol. Now this inositol can be phosphorylated reversibly at all of these positions, giving rise to 7 different species of phosphoinositides that are color-coded here, and that are differentially distributed in cells. And we and others have shown that these phosphoinositides essentially tell cellular membranes who they are. So they're a passport. The red lipid here, PI45P2, marks the plasma membrane, whereas the blue lipid here marks the Golgi. This is PI4P. So in a fed cell, PI4P, a lipid which is phosphorylated here at this hydroxyl group with 1 phosphate, just 1, is essentially a marker for anything that leaves the cell on the circulatory pathway, including the Golgi complex. Now, PI3 phosphate, so a lipid that gets phosphorylated here, is marking the endolysosomal system in a fed cell. That is all you need to know for now about these lipids. So they're differentially distributed in a fed cell. So is there perhaps something that's connecting the changes at the level of the Golgi and the level of the lysosome that explains what happens in a starved cell? And so what Michael did is he used lipid reporters that can selectively report on the lipids, on the lipid membrane content of Golgi and the lysosome in cells, in live cells in this case, and then compare what happens when these cells undergo starvation. Now, let's go through this easily. We're looking at PI4P here, right? So the lipid that is in a fed cell marking the Golgi complex. Giantin is a Golgi marker, and you can see that PI4P marked by this reporter here is mostly at the Golgi. If the cell undergoes starvation, that marker is not completely, but is partially depleted from the Golgi complex, and this can be seen quantified down here. If you look at the same lipid at the level of the lysosome here marked by a dye called lysotracker, and you can see that most of the lysosomes in the fed cell, although there are a few that have PI4P, mostly are PI4P negative. Now, if you starve the cell, the lysosomal PI4P gets ramped up. So with these and many other experiments that I don't have time and I don't want to bore you with, we came up with the following scenario that I would now like to take you through in terms of experimental evidence and what it means. In a fed cell, PI4P is marking the Golgi complex and essentially allows cells to secrete whatever it needs, including growth factors, receptors for those growth factors, and many other things. At this stage, lysosomes actually hardly ever contain any PI4P. PI4P in cells is consumed through a phosphatase that in fed cells is located on the endoplasmic reticulum and partly on the Golgi, called SAC1. Now, SAC1 through membrane contacts hydrolyzes PI4P that eventually shows up on lysosomes because they directly touch. If you go through from a fed cell to a starved cell, we see a prominent change of the localization of these enzymes. a kinase that selectively makes PI4P, normally at the level of the Golgi, called PI4 kinase 2 alpha, gets relocated to the surface of the lysosome, where it can make PI4P, whereas the phosphatase that normally sits on the ER and counterbalances any PI4P that is made on the lysosome is relocated in part to the Golgi complex. So the net effect is that your cell gets rewired. The content of lipids that mark the organelles is flipped from an anabolic organelle, the Golgi, to a catabolic lysosome. So the hypothesis then really was, what follows from this is that through a pathway that I will explain to you in more detail, cells are able to sense the nutrient status, and depending on the nutrient status, they either have lysosomes that are mostly anabolic and growth promoting, and these are the sites where nutrient signaling complexes like mTOR are active. While when cells undergo nutrient starvation, lysosomes actually become catabolically active through loss of the lipid mark that is present in the fed cell and acquisition of a lipid mark that is selective for the starved lysosome under conditions of nutrient deprivation. Let's see how that pans out. So we use live probes to actually look at individual lysosomes, and I brought you one example here. The movie goes around in circles. It starts with a lysosome. Let's go back. It comes back in a moment. That starts out green in a fed cell. Then we acutely deprive this lysosome, the cell, of nutrients, and you can see the green is converted to purple. This is marked here with a dye that reads out lysosomal proteases. So within an individual cell, you can watch the same organelle moving from a growth-promoting into a catabolic organelle. You can play this game now in various different ways, and I will spare you the data. You can take out the kinases that make this, and this gets blocked. You can play with the phosphatases. We've generated knockout cells. You can do acute manipulations. You wouldn't care. The message is apparently the lysosomes change. So what is the mechanism of this change? Well, one element of this mechanism we found out through a technique called proximity labeling proteomics. What you do in this case is you express a membrane protein that is present on lysosomes that carries a promiscuous biotin ligase, in this case APEX2, ascorbate peroxidase, and through an acute labeling just for 5 minutes, you compare the proteome on the surface of the lysosome in a fed cell and in a starved cell. And some of the changes that we saw when we actually conducted this experiment was that when cells undergo starvation, or a pharmacological mimic of starvation, they actually start to acquire lipid phosphatases that degrade the lipid that is characteristic of the fed anabolic lysosome PI3P. You remember, the green one that was on the movie before, right? So MTMR14 degrades PI3P and in fact gets recruited to lysosomes in a starved cell, while the kinase that makes PI3P gets depleted. There are other changes that occur that we can discuss. Now, MTMR14 is in fact an interesting enzyme because mutation of MTMR14 is known from human genetics because MTMR14 is causing a disease that is a myotubular myopathy. And this myotubular myopathy is essentially fatal in children because it results in a failure of the muscle syncytium to properly form. The kids that are affected by this are wheelchair afflicted usually. They're ventilation dependent because they're just too weak to breathe. They usually die at a very early infant age. Now, if you deplete cells of MTMR14, the lipid switch that I just showed you is abrogated, and as a result, the cathepsin activity that normally gets ramped up in the starved rhizosome actually fails to rise to the same level. It's not a complete block, but it's a partial block. And in fact, you can also rescue the effects of PI4P accumulation on lysosomes with respect to position. You wouldn't care about that too much. So can we actually prove, can we understand the lysosomes that are present in a fed cell and in a starved cell in an ultrastructural level? All of what I've shown you now is sort of correlative biochemistry and light microscopy. But can we actually find the organelle, and can we look at it? Can we portray a molecular picture of it? And so in order to do that, Dima Puchkov in the lab conducted correlative light and electron microscopy using actually markers and probes in which we can mark lysosomes through a live label. Lysotracker is a sensor for the lysosome lumen. We can then co-express lipid markers that are selective for either PI3P or PI4P, and we can compare fed and starved cells in movies. So what you see here are lysosomes that are dancing around. You can see they're wiggling. Some of them are static. The nucleus is somewhere here. And then what you can do is essentially you fix into that movie, you embed the sample, and through fiducial markers, you can then reconstruct the entire cell through focused ion beam scanning electron microscopy. So that allows you to do a volumetric reconstruction of the cell and to find back that particular dot that you were interested in. And you know now precisely, because you can use these markers, what the molecular signature lipid of these lysosomes is. And if you do that, you essentially find that cells have two types of lysosomes. The one type of lysosome is the one that was portrayed in the 1960s by Christian de Duve, who got the Nobel Prize for discovery of the lysosome. These are degradative organelles. They're very electron dense. They're perinuclear, they're immotile, they're very highly acidic, they've got a very high content and activity of lysosomal proteases. They do not contain mTOR, or very little. They're often ensheathed by the endoplasmic reticulum, which is involved in lipid transfer between lysosomes that degrade material and ER that makes lipids. And these same things accumulate if your cells undergo starvation. So this is the degradative lysosome that you know from textbooks that has a lot of protease activity. Now, the other type of organelle you also find are these guys here, and they don't quite look like typical lysosomes. They look like multivesicular endosomes. They're much smaller in size. You can see here the diameter is about half, which translates into about a 5-fold difference or so in overall volume of these lysosomes. They're very highly motile. They're typically in the periphery. They're wiggling around. They have relatively low lysotracker signal. They're not quite as degradative, and they accumulate in fed cells, and there is a site of mTOR signaling, as you will see in a moment. So this brings me to the final point of this story. So it appears that cells harbor two sets of lysosomes that are interconvertible. In fact, the textbook of cell biology needs to be expanded at that level. And we propose that the one type of lysosome is the lysosome that Falstaff, a literary figure from Shakespeare's works, used to enjoy when he was drinking wine and eating a lot of food. And that is when mTOR is constitutively active. I can assure you this is not a very healthy thing to do, but it's most comfortable to enjoy. Whereas the hungry or grumpy state here is the state when your plate stays empty, your mTOR is off, your mood might be bad, but you might live for a very long time. So you have to choose whichever you want. So let's look at mTOR, so the coordinator of those anabolic and catabolic pathways. We engineered cells to endogenously express a key component of the mTOR complex called Raptor with an endogenous GFP label. So now we can watch mTOR in cells. Whenever cells are fed and healthy, part of this mTOR, not all, but part of it is located on lysosomes decorated here by LAMP1. If I get rid of PI3P, so the marker of the fed state, mTOR is entirely cytoplasmic. If I accumulate PI4P on lysosomes, so the lipid that is accumulating in the starved state, I also lose mTOR on lysosomes. So mTOR likes PI3P, but it hates PI4P. Okay? So what happens to signaling? You will see exactly the same picture. Low PI3P equals high PI4P, right? So this is the starved state, and mTOR goes off. This is read out here by a substrate, a well-known substrate, phosphoryl-6 kinase, that gets repressed either if you inhibit synthesis of PI3P through this inhibitor, or if you elevate PI4P levels, if you eliminate this phosphatase here that eats it up. You can also play the yang of that yin and induce PI3P. So in the phosphatase mutant that was mutant in the human disease, mTOR is always active. It's hyperactive. And it's also hyperactive if you eliminate the kinase that makes PI4P. So you got very low PI4P, and low PI4P means high PI3P because it cannot get erased, right? So that phosphatase that is mutated up here, you get exactly the same thing. We looked at many different mTOR substrates, at many different cell types. We think it is always true. Perhaps in a slightly different level in different types of cells and tissue. So what I've told you so far is we think we've discovered a nutrient-regulated switch in which the lysosome comes in two flavors, in a yin and yang. The yin in a fed cell is the anabolic lysosome that drives and promotes any anabolic reaction that occurs in your cells that is sensitive to mTOR regulation. Whereas nutrient-deprived cells actually lose this mTOR complex from their lysosomes through an acute response in which PI3P gets hydrolyzed, PI4P gets made, and in this active, catabolically active form, nutrients are recycled that actually keep and sustain the cell alive under conditions when you don't eat. So we think that these pathways, in fact, are also linked through mTOR signaling because the kinase that signals the starved state, in fact, is a downstream substrate of active mTOR. So we have a regulatory pathway in which this state crosstalks with this state, and we've not really understood that in complete detail, but we're really trying to understand that better, how that actually works. So if I've got more time, then I would like to share with you a second story that is very different. Probably you guys are also interested in different things, so I hope you can bear with me for another 15 or 20 minutes on a completely different role of lipids that we're also interested in my lab. And that actually is related to the question of how the brain develops. Now, I'm actually not a neuroscientist by training, so maybe you guys know more about how the brain develops than I have, and you should stand here and I sit down. But let me tell you the level of analysis that we are excited about. Now, what happens in brain development, even postnatally, this is a newborn baby, even with a newborn baby, you can see there is a massive formation of neurons. There is neurogenesis. There is about 100 billion neurons or so, and each of them forms about 1,000 synapses that are characteristic of the mature brain. Now, we know a lot about the programs that actually govern this process at the genetic level, where you have stem cells, and these stem cells receive signals. They go through defined programs. The stem cells differentiate. They migrate to actually form the various cortical layers that are characteristic of our brain, and eventually become mature neurons. But like with all genetics, we take the program for granted. But each program actually needs somebody to execute it. Anybody of you who's ever used a computer program will be able to agree with me, because at the level of the program, yes, it looked fine for the programmer, but the user needs to be able to use and read it. You need to put something into it in order to get any meaningful information out. So something has to happen that actually allows cells to translate the information that is derived from the induction of neuronal gene expression into the formation of a structure that is capable of transmitting and conveying signals between nerve cells. Now, there's a lot of things that need to happen here. This is my poor view of a stem cell in orange here. And that stem cell magically now eventually becomes a neuron, in this case a hippocampal CA1 neuron, and you can see that it's got apical and basal dendrites and a long axon, and there is probably hundreds of synapses formed on the apical and hundreds of synapses formed on the basal dendrites, and then the axon will make another set of synapses which are outgoing with its neighboring neurons. That's all well and fine, and that's cell morphology, but there is also cell biological problems associated with it that go beyond the fact that the cell forms lots of differentiated processes. And that is, the cell needs to form organelles the stem cell didn't have. Now, that's a specific situation because, in all of your skin or hair cells or in the liver that regenerates, the cells usually don't have the problem. The cell just divides. And the cell divides also its organelles. It duplicates its amount of material. The mitochondria get redistributed. The ER vesiculates and gets distributed to the mother and daughter cells. But that's not how this can happen, because the stem cell, even though it may start to induce neuronal genes, it doesn't know what to do with these neuronal genes because it's actually never in its life seen the machinery that is involved in neurotransmission. These are synaptic vesicles that are these yellow balls here that carry the neurotransmitter that get clustered at a presynaptic site that is decorated by active zone proteins that you heard a lot about from Stefan yesterday if you attended his seminar. And these, in fact, direct regulated calcium-dependent secretion at these sites of synaptic apposition. So these organelles need to be formed from somewhere. They don't fall from the sky. Well, you can take it for granted maybe that the gene gets expressed, but how does it go from a gene to an organelle? And this is what we wanted to study, and I think in the interest of time, I will skip this beautiful movie that you can see here from work that we did together with Silvio Rizzoli, which is the sort of Platonic view of a standard mammalian brain synapse where you can see and appreciate the complexity of that organelle. This is the plasma membrane. Here is where the neurotransmitter eventually gets released, and now layers are taken away, and you will see that it's filled with synaptic vesicles and specific machinery that actually enables the synapse to carry out neurotransmission. So how is this thing actually formed? And this is work done by a former PhD student, now a postdoc in the lab, Sila Rizalar, who's essentially tackled the question of how these various presynaptic components, in particular the synaptic vesicle proteins, you can see an accurate reconstruction of such a vesicle here from Reinhard Jahn's work, in active zone proteins get delivered to the synapse to eventually form those organelles. And I can assure you this is sort of a work report. We don't answer that question, right? I can provide you with some glimpse and level of analysis of where we are at this point. Now, the approach we've taken is an approach that is a cell biological approach in which we take human stem cells and convert these stem cells through an established protocol, actually by Marius Wernick and Tom Suter at Stanford, where through the regulated expression of one single transcription factor, you can generate glutamatergic neurons over a time period of about a month or so, actually a little bit longer if you really want to have good neurons. And then this allows you to eventually really result in neurons that make synapses that are functional. So this is electrophysiology. These are extracellular, patch clamp recordings from postsynaptic neurons stimulated here with trains of high frequency stimulation. And you can see very typical signs of fast synaptic depression that is characteristic of these glutamatergic neurons. You can do light and electron microscopy. You can see presynaptic terminals that are filled with neurotransmitter vesicles that are opposed to a postsynaptic membrane. There is one issue with these cultures, I'm looking at Hyo-kyun, because these actually don't form spines in our hands. So these synapses are mostly somatic and dendritic synapses that are not on spines. These guys are very reluctant to make in spines. But by and large, I would say this is a very good system to experimentally study this process. And if I say experimentally study, then I actually mean that we can allow through this differentiation process, we can follow the transport and formation of the synaptic compartment by live imaging. And so what we do is we conduct live imaging of either virally expressed presynaptic proteins or genome-engineered presynaptic proteins. So the stem cell system allows us to do CRISPR and on locus and to have proteins expressed at the endogenous level. And then you can tag them with various fluorescent proteins, looking here at an active zone protein called bassoon and a synaptic vesicle protein, synaptophysin. And these are the movies that you can analyze in the form of so-called chymographs. A chymograph is a line scan over time, so it allows you to see things move. If nothing moves, you get a straight line. If things move anterogradely, you get a line that goes from the upper left to the lower right corner, right? So these guys are moving fast. We then use automated image analysis through a program that we call ChymoButler that allows us to automatically trace these images and then quantify them. And then if you do this, you actually get things like this. So this is endogenous synaptophysin, a synaptic vesicle protein, followed over time. And you can see that about a good half or so of these events are anterograde transport events. There is also stuff that stays and doesn't seem to move over the time. And there is also some that actually move back. And we can discuss about why that may be. We don't care about those at the moment. So the first question we wanted to know is what we're looking at here in these neurons that are undergoing differentiation. Are these new proteins? We want synapses to be newly formed, right? Not stuff that is somehow redistributed. And so you can, again, block protein synthesis. These are two drugs that can do this. And you can ask what happens to your events. And it's very easy to see that, in fact, the red bars here creep down to the bottom. So most of the anterograde transport, if not all anterograde transport events, are essentially gone. So this protein that you see here that is moving to the lower right corner is newly made. So these are our guys that we're interested in. So we did a lot of cell biology, and I reckon you're not cell biologists mostly that are too interested in all the details of this, but let me sum up a few things. So what we found was that organelles, presynaptic vesicle and active zone proteins get co-transported. This is by no means a given, because synaptic vesicle proteins are membrane proteins, and active zone proteins are big cytoplasmic proteins. They're hitchhiking on the membranes. And that makes sense, sort of, because what it suggests is that there is transport packets made that kind of have a preformed presynaptic unit. Okay? Everything you need, well, most of the things you need are together. Now, these axonal precursor organelles do not have markers of the Golgi or typical endosomal markers of recycling endosomes. This is unexpected because they're newly made. They must have come from the Golgi at some point, but they do not contain marks of the Golgi. So they're somehow different. They're a special organelle. Well, they're special in the sense that they contain now something that you now know more about than before, hopefully. They actually carry lysosomal membrane proteins. So the typical stuff that you would say in a normal cell is at least to some degree making catabolism. But obviously they're not making catabolism here because they're building a synapse. But there is no proteases in there. And in fact, they're also not acidic. So these are unique things somehow that built the synapse. That was our conclusion. Now we also, because we're lipid guys, and we love these lipids, we were interested in what is the lipid mark of these. And in line with the fact that they carry lysosomal membrane proteins, they also are marked exclusively by a lipid that in the garden variety cell, you only find in the late endosomal lysosomal system called PI35P2, the lipid here in orange. This is synaptophysin RFP, the axon. You would go in this direction, and this is the lipid sensor, and you can see that actually quite nicely co-decorates with this. You actually also need this lipid because if you eliminate the synthesis of this lipid either through a drug that kills the kinase activity that makes this lipid called apilimumab, the red bar goes down like in cyclohexamide, or you can knock out that kinase, and these are 3 different knockout clones, that bar also goes down. So that lipid is required for axonal transport somehow, and you would want to keep that in mind until I finish. So it's a special organelle, and we were kind of excited about the fact that it is a special organelle. So what does it look like? Now, that's a real tough nut to crack. In fact, if you go through the literature, there is some papers from the late 90s you can find from Craig Garner's lab that were purifying vesicles from the rat spinal cord, but you don't really know what you have there. And then there is this very old study, more than 40 years back, from Tsukita and Ishikawa, in which they ligated nerves and essentially then did EM proximal to the ligation site to accumulate vesicles that they interpreted might, of course, carry stuff to synapses. They didn't know. And this was stuff like this, a heterogeneous population of vesicles that you can see here in this sciatic nerve that is ligated. So we wanted to know what is the ultrastructure of these. It's a very difficult task because these neurons are grown on another cell layer. And it's very difficult now to find these tiny vesicles that are carrying the presynaptic cargo. So what we did is we did a chemical trick in which we hooked up our synaptic vesicle cargos with a protein that allows us to acutely recruit these vesicles in the axons to mitochondria. Now that sounds like a crazy idea, right? Why would you do that? Well, you would do that because I can always find mitochondria. Very easy. Anybody of you can find mitochondria in an electron micrograph. And now you can compare the presence or absence of rapalog and see if now any suspicious stuff in a live clem experiment, again, so you're watching these things, you're adding rapalog, you see vesicles being recruited, and then you embed these samples for Fib-Sem milling. And if you do that, you actually get vesicles hooked up. This is the mitochondrion, the red thing here, right? So you get organelles like this that get hooked up to the mitochondria, and now you can characterize these vesicles. You here see some examples in 2D sections. And what you get is the following. Most of these vesicles are relatively small. This is here in this cumulative plot. About 60% or so are around 60, 70 nanometer. Okay? Now, there is also some larger organelles that we can talk about that are sometimes elongated, like this one here. This one is probably an artifact. And you can see they're sort of dribbling up here towards longer length scales. And we don't know why that is, but it very nicely matches what Tsukita and Ishikawa saw in 1980 in the ligated nerves. So we think the presynaptic vesicle precursors are these kinds of organelles. These are the vesicles that make a new synapse. So what can we learn about them in molecular terms? We had shown earlier together with Stefan Siegrist's lab in this paper that in the fly, when we eliminate a small GTPase that in a non-neuronal cell traffics lysosomes anterogradely on microtubules called RL8, in fact impairs presynapse assembly. So we were speculating that a very similar machinery and probably additional molecules must operate in a mammalian neuron. So we eliminated RL8. We actually have 2 genes of that. It's an essential locus, so if you don't have RL8, you're dead. If you eliminate RL8, you can see only straight lines. So presynaptic vesicle proteins and active zone proteins are no longer transported, or very reluctantly. This is specific. You stick back in one of the RL8 variants, and you get back transport. We also then conducted quantitative proteomics of RL8 interactors, and what we found were essentially 2 sets of things. Synaptic vesicle proteins. Well, that's a great finding because that is what is not transported when you don't have it. And the other thing we found was essentially different kinds of kinesin in addition to some lysosomal proteins. And the one I would like to focus your attention on is KIF1A, because KIF1A had been implicated through genetics in this pathway. KIF1A is a kinesin that is, in fact, linked to neurological disease. There is a specific term for kinesin KIF1A-related neurological disorders, and they all result in some form of paraplegia, which is a peripheral nerve degeneration that in particular applies to the legs and arms and all the lower limbs. There's also brain phenotypes in those. So you got a real problem if you got mutations in KIF1A, and there's actually a huge armada of mutations, some of which are depicted here. Now, KIF1A also got a lipid binding domain that in C. elegans had been implicated in binding to lipid marks and to phosphoinositoids. So we were interested in studying the function of KIF1A, its in relationship to the pathway. So first of all, if we knock out KIF1A, anterograde transport is blocked. It's a specific phenotype. You stick back in the wild type, it is restored. If you stick back in a mutant that doesn't have the lipid binding domain, it doesn't work at all. If you look at the level of synapses, in fact, postsynaptically, these neurons are perfectly normal. Homer is a postsynaptic marker. Presynaptically, you got a problem. Bassoon is an active zone protein, and bassoon is missing in the KIF1A. And if you look at the number of synapses where pre- and post-synapses are opposed, you actually don't see them, or you see very few of them. This is, again, a specific phenotype. If you try to do electrophysiology from these, and we tried hard, we got no responses whatsoever. So that's probably amplified through the neuronal network. So how does this kinesin now, and you see more of these mutations, recognize the machinery? So we knew that on the one hand, this molecule can bind to the small GTPase, but we were interested in the phosphoinositoids. And so what we speculated was that perhaps it is this lipid binding domain in the motor protein that actually reads out the mark on the precursor vesicle. And lo and behold, what we found was that this pH domain very specifically associates with this orange PI35P2, the marker for the late endosomal lysosomal membranes. And if you put in a point mutation, so we did some structural in silico work on this. If we introduce a point mutation in the pH domain that abrogates lipid binding, I don't show you that. In fact, the KIF1A no longer is able to find the precursor vesicles, very similar to when you deplete the lipid, right? So no lipid made, KIF1A doesn't see the precursor, pH domain mutated, it's the exact same phenotype. So finally, we actually wanted to know what is the effect on synapse formation. And in fact, what we saw was that loss of PIK5 recapitulated in part. The phenotype is a bit weaker than that of RL8 and KIF1A, and we can again discuss about that. We saw that synaptophysin delivery to synapses was abrogated or was reduced in a very similar phenotype. This is work with Stefan. In fact, with RNAi, you actually see for the vesicular glutamate transporter is a presynaptic vesicle marker in the fly. So I will wrap it up here. We think that we've discovered a new organelle. This organelle is an organelle that we think is made somewhere in the soma. We don't know how. It's carried along microtubules towards forming synapses. Perhaps gets changed on its way. We don't know. That's one hypothesis you can make about why they're heterogeneous. We've identified machinery that does this, a kinesin, a small GTPase, and a lipid molecule that as triangular mark serve to direct these vesicles to forming synapses, and we think this mechanism is evolutionary, highly conserved. So where do we go from here? That is just an outlook of where we want to go with this. Well, we have now started to get a glimpse into how the synapses form. How are actually synapses now built from the delivery of these vesicles? We don't know. Are they fusing with the axonal plasma membrane? Or is there further remodeling going on? There is some presynaptic components like calcium channels that don't seem to use that machinery, so those have to get there. All neuronal synapses built the same way. You could always say, well, this is a cell biological system, this is an artifact of in vitro, blah, blah, blah. It's probably going differently if you go to a real mouse or animal. So we need to learn more about other types of neurons. What we are very interested in is the question of whether, a developing neuron, the making of a new synapse, and the maintenance or building of a synapse in an adult is via the same machinery or via different machineries. And that's because synapses are not completely stereotyped, they're different, but the same type of neuron will have within a range, a certain number of vesicles in active zone proteins. So a CA1 synapse will have 250 vesicles or so, but never 5,000 and never only 10. So there needs to be a feedback regulation in which the synapse actually crosstalks with the soma to tell, okay, we're good. I know it just need to maintain, but being good is not good enough in the brain because you also, over decades, need to have mechanisms that retain your memory that was formed when you first entered school or your first kiss or your baby was born. You will store that for decades. You can go to PubMed and ask what mechanisms do we know of that. I think nothing. So really, how do you store information over decades? We speculate might involve pathways like this to reset a new steady state, or as computational neuroscientists say, recompute synaptic weight. So with that, I will stop. I thank you very much for listening. I hope you got something out of it, and these are the great armada of people. Wanjol Jang did the initial work on the ER. He's moving back to Korea. Michael Ebner did the lysosome work, and Sila Rizala, together with Max Lucht, did the work on the precursor vesicles. We've enjoyed wonderful collaborations, last not least also with Stefan Sigrist and his lab, who's sitting in the front row. And these are other people who contributed, various other collaborators, and the sources of money. Thank you very much.
Thank you so much for energetic and wonderful talk. Is there any question? Okay.
Are you all shocked?
Oh. Very impressed.
So, in response to nutrient signal, how do the PI K2 relocate from one organelle to another organelle?
You're talking about the lysosome story. So it's a very good question. So we actually don't know what recruits PI4K2 alpha to the lysosome, but we know that in a fed cell, there is a repressive signature put onto it, which is a phosphorylation that is affected by S6 kinase, which is a downstream target, an indirect target of mTOR. And in fact, it's a single phosphorylation site that changes between fed and starved. And when we mutate that site, in fact, we get a constitutive localization of 2-alpha on the lysosome. But how it really, what it sees there, we don't really know. It's actually a good question because it's a fairly simple protein. There is a kinase domain, and it's palmitoylated, but that is presumed to be non-selective. And there's not so much else in sequence. It's been shown to associate with some lysosome-related adapter complexes. Maybe that is contributing to it, but we would like to know more.
Okay. Is there any other question?
Thank you. You're very kind. Clearly in cell biology, sorting is a big issue, right?
Yeah.
So how do you envision that in the formation of these novel organelles that some proteins are excluded, but clearly the ones that are important for biogenesis of the synapse are included?
That's another very good question. So in C. elegans, and mostly in C. elegans, in fact, AP3 adapter complexes have been implicated in sorting axonal cargo. That can't be the sole mechanism in mammals because AP3 is a non-essential complex, and in fact, synapses form relatively unperturbed in its absence. Possibly there is redundancy. There is sorting signals have been identified on synaptic vesicle proteins. Whether and how those relate to it, I think at present is unclear. We're doing a screening approach at this point where we're trying to reconstitute that system in a non-neuronal cell to really go through essentially the entire sorting genome to see how that is done. We think also that perhaps cycles of exo and endocytosis may contribute to sorting, at least at some level, either in the soma or more likely later in the axon.
Thank you.
So maybe two questions. First one is related to Ian's question. So the mark of the lipid is kind of targeting signal by other proteins, which is switched by the condition of the cell, right?
Right.
What is the initial signal I may missed? Is this mTOR, which sense the nutrient, then convert the switch? What was the initial input?
Yeah, that's another good question. So, we haven't completely untangled the you're asking about the upstream nutrient signal that is really sensed. So we know that in the case of the lysosome, surprisingly, it is not the extracellular amino acids that are sensed, but it's a component from the serum. We suspect it's not the growth factors, but the lipid content of the serum. I didn't tell you much about it, but lysosomes and lysosomal mTOR signaling is very sensitive also to the cholesterol content. So cholesterol and perhaps other dietary lipids could be instructive for this particular mechanism. In the case of, which I showed you only in passing in the beginning, of the ER to mitochondria pathway, we know it is the amino acid signal, and in particular glutamine. But it doesn't seem to be at least the major driver here. That's something we need to actually study in more detail. Whether that's only via mTOR is also something I don't know. mTOR is contributing to it, but it may not be the only thing that's happening.
So the second question is the later part. So kinase inducing PI35P2 has to be in Golgi, right? Is it?
Actually, we see that kinase. I didn't show you the data. So we genome engineered the stem cells to endogenously tag PIK5. And in fact, we find that PIK5 on the precursor vesicle. So that kinase is co-transported with the cargo. And in fact, we also see the PIK5 when we do proteomic labeling of the cargoes. We actually find, we actually see PIK5 associated with KIF1A.
I see.
So we think it's on the organelle itself,
I see.
not in the Golgi.
After assembly of presynaptic protein get together, then this cluster includes the kinase so that they
That's
know where
the
to
idea.
go. Ah, I see.
That's the idea.
Is that general rule? So I was suspecting that they have to have like quality check or quality maintenance mechanism to ensure that where to go.
Yeah, I think you're raising an important question that's related to what David actually asked. There has to be quality control. That's true. The precursor organelle is not perfect in the sense of delivering really a ready-to-go presynaptic compartment. So one of the things that I didn't show was that it also contains at least a fraction of lysosomal membrane proteins, so like LAMP1, CD63, at low levels probably. And these are not meant to be part of the synaptic vesicle. So, and then there is some presynaptic proteins that apparently are not carried in the same precursor organelle, including calcium channels. So there must be at least one other transport organelle that's going out to the synapse. And I think there needs to be a second sort of quality control step to really generate the true synaptic vesicle. The synaptic vesicles clearly are different from the precursor organelle in the axon. That was also a claim made in the past, by the way, that the cell actually makes synaptic vesicles already in the cell body and then ships them out. That doesn't seem to be the case.
Thank you very much.
Any question?
Don't be shy.
Okay, go ahead.
Yes. Yeah, thank you, sir. We really enjoy your talk. Just a rather naive question because I was studying something about hyperactive TOR1 allele in yeast. However, just wondering, especially for the lysosomal studies, like when we're talking about the relationship between TOR and PI3P, PI4P, is there any, how should I put it, is there any experiments that your lab has conduct about what will happen if in those cells TOR1 is now hyperactive, or like, I believe hyperactive will means that they are resistant to rapamycin treatment and perhaps starvation conditions, and what's the possible consequences?
I think it's a very good question that you're asking. We haven't studied it in great depth, but we've gone through conditions where TORC is hyperactive, and in fact, under those conditions, this PI3P to PI4P switch fails, and these cells at least have less PI4P on the lysosome, and are probably catabolically less active. How severe that phenotype is, we haven't really studied in detail, I must admit. But hyperactive mTORC signaling is associated with loss of degradation. Actually, we had worked on a kinase that also operates at the lysosome in a starved state that, in fact, has those two phenotypes. You get hyperactive mTORC, you get a repression of autophagy, and you get less degradation of material in the lysosome when that kinase is inactive. So I think that is what you get. Whether mTOR is the only thing that switches, I think I would be a little cautious at this point. Thank you for the question. Great question.
Excellent talk. I have a naive question. When you do the manipulation in the second part, you didn't see a defect in the postsynaptic side.
Right.
Have you been able to look for the organelles in the dendrites? So using the same way, say whether it's like independent of this PI352P, or it's have redundant pathways, or less requirement because dendrite is not that long. You don't need to.
It's a very good question. What we've done is a characterization at the level of the endpoint delivery, not live imaging, but endpoints. And essentially, we've looked at postsynaptic NMDA and AMPA receptors, and those seem to be delivered normally. What we've also done is an experiment I didn't have time to show you, in which we've looked at calcium signals. So we've used either pre- or postsynaptic GCamp, and that actually it's a bit complicated, that experiment, to understand, because so what we did is we put GCamp either on the presynaptic or on the postsynaptic side at the level of the receptor. And if we then stimulate, we actually see that presynaptically we get a perfectly normal signal. Postsynaptically, we see essentially no response. Our interpretation of that is the problem is in between. It is sort of an indirect evidence in our view that, in fact, there is no neurotransmitter released that elicits the postsynaptic calcium signal. That doesn't exclude that postsynaptically, and that's why I'm mentioning it, there could be defects that we actually haven't really looked at. Postsynaptically, this model is so-so, I would say, because these guys are very reluctant to make spines. Actually, we've now gone through experiments where we've cultured these cells for over 100 days, and then we actually start to see spine formation. But, experiments that go over 120 days are not postdoc friendly. So we're reluctant to pursue that level with that system, at least. What we do actually, we didn't target the kinase now in a mouse model, but we just recently got knockout mice that actually lack one or both of the AL8 isoforms. So those would probably be a good model to address those questions. In the AL8? No. In fact, mitochondria are transported absolutely normally. We use that as sort of a standard control. In the human neurons, the axonal transport of mitochondria anyhow isn't overwhelming. A lot of these mitochondria are lazy guys.
Is there any other question? Let's say thanks to Professor HAUCKE for his wonderful talk.
Thank you for the questions and the discussion. And if you're interested in joining us, Berlin is a wonderful city. This is some views of Berlin, and this is our institute. Drop me an email. Thank you.
Actually, I went there. Actually, this is a really good institute, and this institute has several buildings, and you can visit whatever you want. Okay.
I didn't pay him for that.